So Ken's the Chief Strategy Officer and Head of AI at Omnia Bio, and he's going to talk to us about his group's efforts to implement AI in manufacturing of therapies.
Nice to be here. I appreciate CBTN inviting me back. I have talked here, I think, once or twice before in the past. Just to give you a little bit about my journey with CBTN and then starting all the way back with DIPG. It was about 3 or 4 years ago that Dr. Ash Davison, who worked with me at Amazon, and Dr. Rod Tarago, also worked with me at Amazon, asked me to come to Washington, D.C. to meet with Amanda Haddock and Lisa Ward. I think it was 4 years ago. Was it 4 years ago? Anyway, time flies. So to get interested in a pediatric program, that if I could engage Amazon to think about contributing in some way or another. So I came down, and I have to tell you that from that dinner, the first meeting, this program had my heart, and it's kept my heart even after leaving Amazon, and I'll go through that in a minute of where I am and what I'm doing. It is a focus and mission for me every day to think about what we can do in this pediatric space. So thank you very much for the invitation. I'm really pleased to be here. I'm going to talk today about using artificial intelligence and data and also controlled manufacturing so that we can think about actually increasing accessibility of the things we're developing in this pediatric space and getting more information out of not only our clinical data, but also our manufacturing data in the process. A little bit of background. Yes, I joined Omnia Bio. Omnia Bio is a contract development manufacturing organization primarily focused on rare disease. And if you can imagine what a CRO is and what a pharma company is, we sit in the middle of that. So we do both development work under contract for pharma, and we also ultimately manufacture the final drug product that we did the development work on the pharmaceutical company's behalf. I started this journey, as I mentioned, I was at AWS for 5 years, leading the academic medicine team, and I just left there in June to continue my passion of something that at Omnia Bio we came to CHOP a few years ago and started talking about a broader collaboration in bridging data from CBTN and to start to build analytics around it. And I'm going to give you some update on where we are on that and kind of where I think we should be going. But just to do a quick flyover of, I think the headwinds, and you all know this because you face this every day in your business, but the headwinds really are in brain and CNS tumors. It is just not, it's very hard to get the attention of pharma. Because if you look at these based on the bubbles, this is a McKinsey report, and in the red boxes are the brain tumors. But you see the very large bubbles, those are the other economic interests that pharma has to go after, much, much more profitable, so to speak, therapies. So we're kind of in this together. It's the academic organizations and really our organization as that rare disease developer, where we're going to have to focus and to solve these problems because big pharma is not going to come to our rescue. And that's what really the basis of my talk is on how to think about it. When you look at the different types of pediatric solid tumors, you know, it's all over the place. But one of the things I love about CBTN and I think we can focus on is if we dial in on DIPG first, there's a data set there, consented patients with most of them do receive some sort of immunotherapy, and that probably it's in a very high priority basis. It's either first or second line treatment. And there's an element of data there that we can do a lot with. And that's basically my pitch today to talk about what can we do next in that space. You know, these are the interventions that are available to clinicians when they're diagnosing brain tumors in the patients. In the very small upper left-hand corner, that was supposed to be bigger, but when it got translated to Google from Microsoft, it shrunk it down. But there are a number of things that obviously you understand in the clinical field that you go through to decide what is the right therapy, et cetera. I'm going to show you some data from our group that actually shows that what we probably thought before, that the patient might be really, really important in this equation. I'm going to show you that actually the donor cells are the most important in the equation for efficacy and treatment outcome. So focusing in on therapies as a whole, this basically just further exemplifies what I just talked about. This is pharma's interest. Brain, again, is in the red box. Not a whole lot of interest or things out there in the pipeline for this space that we're talking about. So we're going to have to do this on our own. And the first area we're focusing on as Omnia Bio is the cellular therapy path. Let me give you a little bit of background of Omnia Bio besides just being a CDMO. So we are backed by the federal government in Canada. We spun out of the University Health Network. So we were a non-for-profit organization at basically around University of Toronto. Come out of that environment and built the first infrastructure for supplying phase one, basically what many of you do in your facility, cell and gene therapies to provide them to the academic environments. We've now grown up since then, that was in 2019, to where we now manufacture all the way through to full commercial drugs for rare diseases. And next week, we'll be cutting the ribbon with most of the politicians from Canada, cutting the ribbon on the new facility to dedicate it where we can do phase 3 and commercial manufacturing in Canada for all of North America. And our goal is to improve access, and in particular in rare diseases. How do we improve access? Well, obviously the therapy has to work, but it also has to be consistent and reliable, and we have to have enough data to convince the regulators to move this from a phase 1 through the process for it to become a commercially viable product. And in that process, as you go from like a phase 1, which many of you may be doing, into phase 2, into phase 3, you get challenged much more by the regulators in what you're producing and in the consistency and the expectation for the clinical trial. And my pitch is here for this business is for us to think about, in particular in CAR T therapies, to be ready to make that evolution and grow out of what we're cooking individually at one of our 30 sites and think about coming together collaboratively now that we have this capability, for example, with us and actually producing much larger batches and throughputs and putting that out into multiple academic medical centers from one drug so that we can get more data and make it more consistent. The other point to what's happening in the industry, and the chart on the very top is funding is drying up for this market. And that's a 10-year, if you look at the bar charts for CAR T as a whole, and in particular for autologous CAR T, the amount of funding that is out there has almost disappeared. And Q2 of 2024 is the lowest venture capital investment quarter that we have seen in 10 years. So not only is pharma not coming to our rescue, but venture capital is pulling out of the space. And we've got to find a way to reinvigorate their interest. And the surprising point behind that is there are over 1,000 CAR T projects globally focused on solid tumors that are gasping for air to get funding to just continue doing what they're doing. And NIH can't do all of those. So we have to look to the private market. And the way we're going to do that, I think, is through consolidation, aggregation of ideas, and being much more focused in our ideas to bring them to market. So ways we think we can do that is, A, we have to get the cost down. So if you understand a CAR T cost just to manufacture it is right now coming out of our facility is about $120,000 per dose in cost. run that through all the markup, and by the time the patient gets it, you're approaching $500,000 or more in medical treatment, et cetera. That's just not going to be, we aren't going to be able to put that out there for the wider audience. We have to get the costs down. We also have to get the product consistency up from site to site to site and really put more drug rigor into it. And finally, this new element that we were not suspecting, but is the fact that the donor variability, so the cells you apheresis from the patient to create the CAR, are highly variable. And we have developed artificial intelligence models that will actually predict that patient's cells, whether that patient will respond to a CAR T or not. And I'm going to show you some of that data in a moment. So we believe by combining picking better patients into the process, meaning that pick responders. If they don't respond, don't spend the money on them, and be fair with family and the patient themselves. But the second element is bringing automation, robotics, and AI into the manufacturing plant and taking humans out of it. Humans make errors. Humans cost a lot of money. And the majority of the cost of where I was talking about the $120,000 is infrastructure and labor. So as we start to think about, okay, we want to put robotics can take a lot of cost out. We want to look at the donor cells, and we want to also understand if we're going to start consolidating manufacturing into a regional area, we also have to think of logistics of fresh samples coming into a manufacturing site, processing that sample, and then getting it back out to the patient in a timely fashion. What we call vein to tumor or vein to vein logistics cannot be underestimated. I was with the top 5 pharmaceutical company 2 weeks ago, and they told me, in their opinion, this is one way most organizations fail when they go to clinical trial, and they're trying to move it around, is they don't have the logistics ironed out. You're sitting here in Amazon today. Amazon knows how to move packages. And if you want to think about being able to solve one of those bubbles very easily, Amazon can do that in their sleep. A single Amazon package goes through 85,000 different AI inferences before it arrives on your doorstep without humans being involved with it. Imagine if we could apply that to biology and medicine, technology that already exists there, in getting that drug out to the patient in time and not needing to freeze it, keeping it fresh from the apheresis through the process and getting a much more viable and potent dose back out to the patient fresh. Could be impactful. But in order for us to do that, we have to have really, really good IT infrastructure. And I think Adam was talking about, the intersection between technology and research and medicine is getting more and more impactful every day. I'm going to introduce you to another factor. And that's the idea that as you think about data management centers and cloud, you also have to think about latency of data and moving it around. We're sitting here in Washington, D.C., which is close to all the data centers running the government. But as you move out of here and head towards Boston, head towards the central part of the United States, or go to the West Coast, which doesn't have data centers, the time to move data and information through those pipelines will not support automation and robotics. The need for a robot to build a drug inside a plant, it has to be under 10 milliseconds of latency from the data center to the robot for it to move and do its thing. Otherwise, you have, awkward movements and vibrations that won't allow us to scale it. So as we're thinking about building in this infrastructure, really only 2 places that can do this. First one is here in Washington, D.C., because the cloud centers are close. And the second one is in Toronto. And in Toronto, both Oracle and AWS have built some of the fastest networks available in the world and have about 7 milliseconds of latency from their data centers to our robots. And so we can actually scale up there and take the humans out of the manufacturing process. And what that ends up getting us is a 50% cost reduction per dose. So just by putting in robotics, taking the same technology, we already can bring that $120,000 manufacturing cost dose to 60. That's impactful for families and payers. Second thing that we're focused on is really looking at, what is the raw material coming in? Well, in the autologous space, raw material is the cells from the patient. It is the apheres cells coming in. And our chief scientific officer, this was a paper that was published in Nature Biotech last year, retrospectively looked at liquid tumors and the treatment of CAR T, so Yescarta and Kymriah. And what we typically do when we apherese the patient is we look at their T cells and we immunophenotype them. And immunophenotyping has been the standard for a very long time. But he went back and he said, I'm going to apply machine learning to this, and I'm going to take the current data set of immunophenotyping, and I'm going to evaluate it. And what he found was that the immunophenotype did not predict clinical response. It did not tell us that the patient would effectively be treated with that CAR T. But if you take the transcriptome from the donor cells, basically you sequence the RNA, that was absolutely predictive. And on the chart on the left-hand side, along the very wide colored bands are the clinical response. And along the middle is the machine learning algorithm that we developed to predict whether the patient would respond or not. The most important thing is to look at the blue. The blue areas are patients that were non-responsive to CAR T. And we could have predicted not only their response rate just by running their cells in the machine learning program up front, we could also predict the burden of the tumor over time. So the correlation of the AI program to response is about 88% with machine learning, and in traditional immunophenotyping is under 60%. So a radical change in the clinician being able, right in the electronic medical record, to run an AI model and predict whether this is the right therapy for that patient. So we think this is groundbreaking. We're going to continue to work on this and bring this into the solid tumor space. Has many more variables than liquid tumor. We're talking about tumor microenvironment. And hopefully this will hold up. And one of the things I want to propose to CBTN is to be on that journey with us to explore the data that you have in your database and see if we can build a similar model that we have for liquid tumors. Can we do that for solid tumors? Other areas, and we have big pharma now interested in our approach with the AI and the robotics, is we can now take nearly a 3-week time from diagnosis and apheresing the patient and then to the patient getting the therapy. We can now compress that into about 14 days. So shave a week off of it by using AI and robotics. And a week for some patients is really significant, really, really significant. And we do that by using new methods in quality control and robots. So we're taking humans out of the quality control lab, where we have to do about 8 different types of tests on every dose that we release. The robots do that, and then we release it out to the treating facility at risk. While we wait on the quality control data to come in, the sample's already in flight to the treating facility. And once it arrives at the treating facility, it's released because we will have the readouts that it's a safe drug to infuse. But we're going beyond that, and I think there was some discussion this morning with Rod's panel, et cetera, on AI. What does AI really mean? What I just described to you in the donor assessment is really is machine learning. It is mathematical algorithms that are used to predict and basically statistically predict the probability of something. Generative AI or large language models or foundation models are quite different than that. They actually take the corpus of information that we have out there, and they only know what we've taught it. It's you think about it sort of being as a brain. We will be developing a brain for the CAR T market that will incorporate protocols, it will incorporate reagents, and it will incorporate all the data that I talked about before that will allow not only scientists and process scientists to interact with it to run what-if scenarios. But it will go all the way back to ideally where we can implement that in Epic as an example and let the clinician run the what-if scenarios right on the generative AI model while they're restaging the patient. So it gives them the power of information at their fingertips, which they don't have right now in that particular space. Obviously, with developing generative AI or any AI models, you have to think about, kind of your guardrails. AI is not to be trusted 100% in what it's doing. It gets confused. And one of the things we have to do in implementing AI is actually secure the AI from the outside world. And what does that really mean? It's like we take a foundation model inside and we cut it off from any of its further training or learning from whoever the original owner is. So if that's OpenAI, if that's Amazon, if that's Microsoft, we need to shut it down from future learnings because someone else can come in and take a trained model and introduce it to new data, and it can chase that data and think that that information might be reliable. Information may not be reliable. So we have to bring it in and lock it down. So first thing, and I would challenge all of you, is to think about getting any machine learning or AI model that you're using, containerize it. Shut it off from anyone else teaching it anything. If you're going to teach it to do something, have it do that activity. And then if you want it to learn to walk, and then you want it to learn to ride a bike, do it sequentially one at a time and make sure once you teach it to ride a bike that it still knows how to walk. And you're just always going back and validating and verifying at each step. And that's got to be within your own organization. You need to securely control who has access to the model. So as we start to think about this and how we can advance accessibility and affordability of immunotherapies, it really takes a village. And on the right-hand side of the slide are who we envision the partners need to be in this ecosystem. And yes, it is academia. It is the AMCs, et cetera. It is big tech. We cannot do this without big tech. As I showed you latency, we can't do it without their data centers. And yes, it's ultimately going to be pharma because pharma is going to invest in the robotics and the capability that we're going to learn from that we can't afford to do on our own dime. And it's going to be governments. So if you look at, for example, for us in Canada, the Canadian government is making a massive investment, not only into artificial intelligence for the country of Canada, but for us as a rare disease biomanufacturer. They are making, they're going to head into their second round of investment to give us the belt and suspenders we need to execute on this, not only for the Canadian market, but for the globe. And having that kind of underwriting and support is critical to be successful. So with that, I'll just kind of conclude, but really my appeal to you is think about syndicating, think about working with an organization like ours and what we can offer to you. You have the intelligence, you have the know-how, we have the capability to execute on it and make it consistently and put the robotics and the automation in to get your costs down.
Thank you very much, Ken. Any questions for Ken? See a few already, but I'm going to...
Before you go, because you might be able to answer this question, I thought you were going to scoop us for the question, Chris. Okay. It's actually a question for CBTN because especially with the event this week and so many external companies coming, you don't need to answer this right now, but I also get a lot of questions of what does your approval process look like for decision making? What's that governance look like? I've been with Allison at certain executive board. I think that governance of CBTN will be helpful for external folks so they know how to help you move faster. And I don't think I have that in my head. So
I think it's a great question. Maybe it's tangential or related, but it sounds like in Canada, like this is really moving forward. What have your discussions been on the US side? And what would it look like to do a US-Canadian partnership or framework?
Yeah, I mean, I think that for us to think about this syndicate, we're going to need some US government backing and funding for it. So one of the things we've done as Omnia Bio is we just incorporated in the US so that we can check that box for NIH and others. You will also see some potential collaborations coming out from academic medical centers that will be meaningful in that area as well. And we're really excited about that. So it's where we will join together to form joint ventures to do this work and go get those grants. So the Canadian government's going to underwrite us for what we can do within Canada. But I think we have to rely on the US government to underwrite us for what is going to come out of here. But it doesn't mean that we need to rebuild the robotics system in the US because we can easily let everything flow back and forth. And it is a tax-free movement. So we don't have barriers at the border like you would have if you were sending it to Europe, et cetera. So the US, Canadian, Mexico Act, which replaced, replaced what was the NAFTA before, allows all of these systems to go tax-free and to flow through automated port arrival and departures. So I would say for us, really the most important part is getting the collaboration underway and really think about how we would fund retrospective or prospective trials to look at data to answer bigger questions. And I still have in my mind, and I don't think anyone can answer this, is manufacturing even important? Variability in manufacturing important to clinical response? We don't know that. We should find that out. That's important for us.